No, I'd actually turned those on when I first came in. No.
Perfect. We're ready to go.
Okay. So what we're going to be doing with the rest of this week and next week is looking in detail at what the mitochondria and chloroplasts do in cells, basically harvesting chemical energy by, in the case of the mitochondrion, converting chemical energy from one form to another, and in the case of the chloroplast, converting light energy or electromagnetic energy into chemical energy for the cell to use. These two chapters are probably the most difficult that you'll read over the course of the term, mainly because they have a lot of biochemical pathways. You have to sort of count atoms to make sure, to keep track of where everything's going. What I've done in these slideshows is tried to make it as simple as possible. I've got a lot of additional figures and flowcharts in here to try and help you not lose the forest for the trees. So don't drown in details. I'll let you know what you need to know for the exams out of this. So we start with Chapter 6, how cells harvest chemical energy, and we're primarily concerned with the mitochondrion here. Some of the major points are that cells obtain energy by capturing it from food molecules. We're talking specifically about animal cells and animal-like protists here. And indeed, fungi do the same thing. They cannot process sunlight, so they have to capture it from food molecules or from other sources. Right? That's the job of the mitochondrion. There are a number of metabolic pathways that will be described in this chapter, and that's just to define a metabolic pathway. We can just say it's a sequence of reactions for some common process. For example, the set of reactions that are used to break glucose down into carbon dioxide, that would be a metabolic pathway. A pathway that takes two carbon compounds and makes lipids out of them would be a metabolic pathway. Those are two different kinds of pathways, though. The glucose breakdown pathway would be a catabolic pathway, and the pathway of assembling a lipid from two carbon compounds would be an anabolic pathway. All right? Both of those are used by cells. Much of what we'll be talking about here are catabolic pathways. And this is perhaps the most important point of this chapter. The ultimate goal of the catabolic pathways we'll talk about in this chapter are to make ATP. Okay. Why? Because ATP is the primary energy carrier. That chemical energy in ATP can then be used for a huge variety of cellular processes and functions. And so it's absolutely critical for the cells to not run out of ATP. Right? The cells must always maintain enough ATP. So that's the ultimate goal of all of these catabolic pathways that we'll look at. High-energy electrons are also used in these pathways. Right? We'll see a situation where electrons are being snatched away from other molecules. In a sense, the energy that's in those electrons is captured and stored for release later. Okay, so high-energy electrons are going to be an important part of these catabolic pathways. These energy harvesting pathways require oxygen, and they produce carbon dioxide as a byproduct, which will help explain why we have to breathe in oxygen and why we breathe out carbon dioxide. It's because of all of the these catabolic pathways going on in the mitochondria in all of our cells. That's where the CO2 is coming from. Okay, so production of ATP by using food molecules, high energy electrons, and that way the cell has plenty of energy. All right, so why do we breathe? Well, we breathe in oxygen, we exhale CO2. The reason is because these energy harvesting pathways are breaking down carbon chains. What you're doing is you're going to take glucose, basically, and other carbon compounds, and break those carbons off, generating carbon dioxide. When you do that, the energy in those bonds can be captured, either as high energy electrons or in some other form. Right? But the ultimate point of that is that that energy can be captured. And so when you break those carbon chains, you capture energy from them. But it generates carbon dioxide, which we have to get rid of. We can't use it. Right? So we have to get rid of it. So we call these pathways cellular respiration. Just as in the respiration that we normally think of, breathing in oxygen, breathing out carbon dioxide. Cells are respiring. They're taking in oxygen, using it, they're generating CO2 and getting rid of it. So it's cellular respiration. Okay, so for example, we have muscle cells that are doing aerobic exercise. Right? Aerobic means with oxygen. Anaerobic means without oxygen. Those terms will become important if you don't know them, because we'll talk about two different pathways that cells can use depending on whether they're operating aerobically or anaerobically. So they're breaking down sugar into carbon dioxide and water. They're generating ATP to power the mechanical movement. That's all muscles do, is they convert chemical energy to mechanical energy. You generate lots of ATP. You break the bonds in that ATP, and that allows you to do mechanical movements with your muscles. Okay? The oxygen is supplied by the lungs and bloodstream, of course, and the CO2 comes out the same pathway. All right? So it's sort of a comparison between regular respiration that we think of on an organismal level and the cellular respiration that's going on in cells. This is the balanced equation for cellular respiration. Okay, we're taking glucose, adding oxygen, okay, and sending it through essentially what is now a black box. We haven't looked at the processes involved here. But we generate carbon dioxide, we generate water, and we generate ATP. Okay, other molecules can go into this pathway. Other sugar molecules, for example, fructose, which is a 5-carbon sugar, can come into this pathway. 3 and 4 carbon sugars can come into this pathway. It's only about 40% efficient, and so we generate a lot of heat. But that's good for us, because that heat is what allows us to maintain a steady body temperature. Okay, we couldn't do it otherwise. As I said, other molecules may be used. Fats especially. One reason that we store energy as fats is because fats are a wonderful energy supply. You can get, out of a glucose molecule, you can get 36 ATP molecules. Out of a lipid molecule, a fatty acid, you can get 121 ATPs. All right, so it's a big, big difference. And for that reason, the most efficient energy storage form for animals is fats. All right, so when we take in extra glucose, for example, we convert it to fat. We use glucose first, however, and the reason is because it's the easiest form to use. It's a little bit more difficult to use fats. You have to break the fats down into 2 carbon chains and shunt them into these catabolic pathways, so it's a little bit more difficult to do. So if you've got lots of glucose around, you use that first, then you start going after the fats. If the fats run out, then you start going after proteins. All right? So if you're looking at cellular starvation, that's the order they do things. They use sugars first, then fats, then proteins. Okay? And if you're using proteins, at that point you're in serious trouble. There are some tissues that only use glucose. The nervous system only uses glucose. Okay? If you're starving yourself, what your body has to do is convert fats into glucose, send that up to the nervous system, the brain, for example, and then the brain breaks it down again. Right? It's a huge waste of energy, first of all, and it's difficult to do when you're not taking any glucose in. Right? So the brain is one of the biggest gas hogs in your body. It uses a huge amount of energy to do what it needs to do. Most other tissues can use glucose as an energy source. Okay? Some tissues don't even need oxygen. They can do things without oxygen, at least for a short period of time. All right, so keep this reaction in mind because everything we go through is going to build on this basic reaction. All we're going to be doing basically is filling in this arrow. What happens when you take glucose and oxygen and break it down into those components? What are the steps involved? I kind of like this table. This is kind of a neat table. It shows the energy that's consumed by various activities. This does not include what we call basal metabolism. While you're sitting there passively, you're using a fair amount of energy. Average person will use about 2,000 kilocalories just for basic metabolic functions, keeping yourself warm, keeping your muscles in good tone, all of that sort of thing. So basic activities will take around 2,000 calories, more or less depending on your metabolic rate. So these are in addition to that. That's why they say sleeping or lying still is at 0 because that's basal metabolism. But there are some activities in here that use a lot of energy. If you're riding a bike really fast, if you're dancing really fast, that takes a lot of energy. Swimming is a really good way of getting rid of calories. Okay? So if you don't like jogging, you should swim or go out dancing. Learn to dance. Okay. So what are the waste products of cellular respiration? If you've been paying attention at all, this should be easy for you to figure out. To answer this question, just keep in mind that equation that I showed you at a couple of slides back. You've got glucose and oxygen going in. You've got carbon dioxide, water, and energy coming out in the form of ATP. Okay? So the answer is, of course, C. Inputs are 1 glucose, 6 CO2s. The outputs are 6 O2s. The outputs are 6 CO2, 6 H2O. And around 34 to 36 ATP. Your book says 38, and the reason it says 38 is it doesn't take into account the few ATPs that are actually used during the process. But I do. That's why I say 34 to 36. Depends on whether you're a prokaryote or a eukaryote. Prokaryotes are actually better at it than we are. Okay, so they get more. And we produce some heat, which keeps us warm. Okay, any questions so far? Good, okay. The energy of electrons. When you have electrons in particular orbitals, that means that they have a certain energy associated with them. The farther away they are from the nucleus of the atom, the more energy they carry. That energy can potentially be snatched away from them, right? It can be released. You can store that energy by removing those electrons from those molecules. Okay? So these processes boil down to one principle. Cells use energy from electrons transferred from organic molecules to oxygen. And they use that energy to make ATP. Okay? So cells use energy from high energy electrons to make ATP. I'll say that again. Cells use energy from high energy electrons to make ATP. Okay? And in case you didn't write it down, I'll say it again. Cells use energy from high energy electrons to make ATP. Okay? And if you still didn't get it, that's your own fault. Okay? This is why oxygen is required in the process. You've got to have some place to pass those electrons off to once you're done with them. Once you've released the energy of those electrons, you've captured it as ATP, you've still got these electrons left over. You've got to get rid of them. You dump them off to oxygen. In doing so, you generate water. That's why water is one of the byproducts. Okay, so electrons are removed from organic molecules like glucose. Eventually, after going through this whole process, they're dumped off to oxygen, and that produces water. We have a special term for these kinds of reactions. They're called redox reactions or oxidation reduction reactions. Okay, so we need to take a side note or sideline and look at what oxidation reduction reactions are. Okay. Oxidation and reduction reactions usually occur in pairs. What we're looking at here is a 4-carbon molecule with, on the second carbon, a hydrogen and a hydroxyl. If we oxidize that, that means that we remove electrons. Oxidation is just the removal of electrons. Okay, so what we see here is that we've now generated a double bond between the oxygen and the second carbon because we've removed 2 electrons. We also have 2 hydrogen ions left over. Okay, those electrons need to go somewhere, and what happens in this reaction is they're passed off to a molecule called NAD, or nicotinamide adenine dinucleotide. Just remember NAD. Okay. Makes it easier. It's actually made from a vitamin, one of the B vitamins, nicotinic acid. But what we're doing is we're going to take NAD, we're going to take those 2 hydrogen ions and those 2 electrons, and we're going to add them to NAD, which is a reduction. Reduction is the addition of electrons. Okay, so oxidation, removal of electrons, reduction, addition of electrons. That generates a molecule that you will become painfully familiar with, NADH. Right, it's the reduced form of NAD. It's a carrier of high-energy electrons. Okay, so oxidation. Electrons are removed. We're removing the electrons from that 4-carbon compound. We're donating them to NAD, and that's the reduction part of this set of reactions. Right? These almost always occur simultaneously, and that's why we call them redox reactions, because you almost always see an oxidation followed immediately by a reduction. So we just combine the two and call them redox. Okay, because of these redox reactions and because high energy electrons are being used to generate ATP in the cell, we have to look at the carriers of those high energy electrons, and there's basically 2 important carriers. NAD is one of these. Okay, it picks up 2 electrons, 2 hydrogen ions come along for the ride, and we produce this reduced form, which is always written NADH plus H plus. We know that the H plus is involved. It sort of follows along with the NADH. Doesn't seem to actually be bound into the molecule, though. All right? So a lot of times I'll just write NADH as the reduced form. Just keep in mind there's that extra hydrogen ion sticking out there somewhere. All right, so NAD gets reduced to form NADH. It picks up high energy electrons. It stores the energy in those electrons. Okay, FAD is another one. This one, the long name of it is flavin adenine dinucleotide. And again, it's made from a B vitamin, riboflavin. Right? It will carry 2 hydrogen ions and 2 electrons as well. And when it does so, it becomes reduced to produce FADH2. So it's easy to see the reduced forms and the oxidized forms for these molecules. Plants use NADPH. It's almost exactly the same as NADH. It just has a phosphate group on it. Right? So that's really the only difference. Okay. The other energy carrier for the cell, of course, is ATP, which we are very familiar with. All right, so NAD, FAD are reduced. They carry high energy electrons. ATP carries chemical energy in the form of phosphate bonds. Right? Why are electrons used? Okay, well, as I've sort of said several times already, electrons carry energy. The energy in those electrons can be released, but it's not released all at once. If you took the energy in those electrons and released every bit of it at once, poof, spontaneous combustion. Well, okay, not quite that bad, but you would burn up. Your temperature would rise to the point where your proteins would not function and you'd die. You'd get a super fever. So our cells release that energy a little bit at a time, okay, so that the heat becomes more manageable. This is occurring in a process called the electron transport chain, and it's happening in the mitochondrion. All right, the electrons are just dropped off from one carrier to the next carrier to the next. And every time they're dropped off, a little bit of energy is released. Right, that energy is then used and made available for making ATP. The electrons are finally dumped off to water. Remember, I said that you've got to get rid of these electrons that you're stealing off of things like carbon, the glucose molecules. You've got to get rid of them eventually. You drop them off to water after they pass through the electron transport chain. And so water becomes a waste product. Here's how it works. This is a summary, diagrammatic summary of an electron transport chain. Okay, we have a high energy electron carrier up here, NADH. NADH participates in a redox reaction where this time NADH gets oxidized and the electrons get passed off to a carrier, typically a protein, right, in the mitochondrion. However, there are other non-protein electron carriers. Those electrons are then passed from one carrier to the next, and every time they're passed off, they lose a little bit of their energy. Okay? So continual passage goes down an energy chain. Think of this as a graph where we're looking at potential energy on the y-axis and the course of the process, time basically, on the x-axis. And over time, you pass off these electrons and you drop in energy every time that you do that. But that energy isn't lost. Right? It's being, in a sense, converted to another form that's allowing the cell to make ATP. Okay? So electron transport. All of those electrons are going through this electron transport chain. How do we make ATP then? 2 ways that the cell uses to make ATP. First type of ATP assembly is in the electron transport chain itself. Okay? I haven't told you yet where the energy from those high energy electrons is going. Now you know where it's going. It's being used to pump hydrogen ions across a membrane, across the inner mitochondrial membrane to be precise. Okay, so now don't lose track of where we've gone. We're taking glucose, we're stealing electrons off of it. That gives us FADH and NADH. Those electrons are passed off to an electron transport chain. They're passed from high energy to low energy. The energy in those electrons is used to pump hydrogen ions across a membrane. Remember from last time, membranes are selectively permeable. Hydrogen ions are charged. They don't get across membranes. Okay, so if you pump them across a membrane with a carrier protein of some sort, they're going to stay across that membrane. What you've done is you've made a gradient of hydrogen ions. And gradients, as you'll remember, are forms of stored energy. Okay? So you take the electrons from glucose, pass them through an electron transport chain, use the energy to pump hydrogen ions across the membrane to make a gradient. Then you let them flow back across, and that is the process that's used to make ATP. That's where the energy is finally released to make ATP. I'll show you a figure in a second that will show you how that actually is occurring. All right. The other process is a simple transfer of phosphate. You can do this too if you have enough energy inherent in a molecule. There are parts of this breakdown of glucose that actually use this method. The molecules are so unstable that you can take a phosphate off of them, just pop it onto ATP. All right, so here is what's happening in the mitochondrion. The energy from NADH, FADH2, those high-energy electrons, pass through the electron transport chain. That energy is used to pump hydrogen ions across the inner mitochondrial membrane. You have a gradient of hydrogen ions, high out here, low inside the mitochondrion. Okay? That's a form of stored energy, just the same as having a dam with a lake behind it is stored energy. Okay? You have a gradient of water. Well, okay, so what you do then is that you let these hydrogen ions flow back through this protein, ATP synthase. And the energy that's released by that process allows that protein to make ATP. It just takes ADP and a phosphate, pops the phosphate onto ADP, and you've got ATP again. Okay. This process is called chemiosmosis. Remember, the term osmosis was the movement of water across a membrane. Right? So chemiosmosis is just the movement of chemicals, ions in this case, across a membrane. Right? So we pump hydrogen ions across the membrane. Those ions flow back down the gradient, release energy, allow ATP synthase to make ATP. The electrons for this process came from glucose and other carbon molecules. This is the other process of making ATP, substrate phosphorylation. You remember the term substrate. A substrate is something that an enzyme binds to, right? So we're just phosphorylating that molecule that the enzyme, this particular enzyme, binds to. All right, so here we have an organic molecule that's unstable. It has a high-energy phosphate bond on it. And you just take that phosphate and you pop it onto ADP, producing ATP as a result. Okay, simple enough. This is used less often by cells, but it's especially important if oxygen isn't available. Keep in mind that the very end product of the electron transport chain is going to be water. That's what we're passing the electrons off to. And we're passing electrons off to oxygen to make water. If you don't have oxygen available, you can't do that. So you do this instead. It's not as efficient, but it gets you some ATP instead of none at all. All right, so there are three main stages of respiration that we're going to talk about here. The first step, glycolysis. Read that as glycolysis. Glyco referring to glucose, and lysis referring to breakage or splitting. Okay, we're splitting glucose. We have 6 carbons in the glucose. It's broken down into 2 molecules of pyruvate, which are 3 carbon molecules. We haven't lost any carbons yet. We've just rearranged the bonds and broke them in half, basically. The next step is the Krebs cycle, or sometimes called the citric acid cycle or the TCA cycle. You see it different terms in different books. Here in the Krebs cycle, we're going to get rid of those carbons in the pyruvates that came from the glucose. They're broken off as CO2, which we exhale. And in that process, as we break those carbons off of this molecule, we capture those high-energy electrons. So this is where most of the high-energy electrons are being captured. It's the Krebs cycle. That's its most important function. The third step is electron transport, which I've spent quite a bit of time talking about already. The energy of the electrons is used to make lots of ATP. Okay, so you take glucose, you bust it into 2 3-carbon molecules. You take those 3-carbon molecules, you pop off all the carbons as CO2s, capture the electrons, send them through electron transport, generate ATP. And that's basically what's going on. Okay. I heard a groan out there. Yeah, I used to do the same thing every time I saw chemical reactions. All right, in the following reaction, which molecule is oxidized? I see a lot of painful expressions out there. Okay, here's a hint. Here's how you figure out these kinds of reactions. Look for a loss of hydrogen ions. You know the NAD is picking up 2 electrons and 2 hydrogen ions, right, to become NADH. It's being reduced. So look for where the hydrogen ions have been lost, and that's where the electrons are being pulled away from as well. So if you look in this, in these sets of reactions, right up here, C3H6O3. On the other side, it's C3H4O3. Okay? Subtle, but that's how you spot these things. All right? See? Right up here, C3H6O3 becomes C3H4O3, and we stick a phosphate on it. Well, that phosphate is really immaterial. Who cares? Ignore it. Right? It's the H4 that we're interested in from H6, where the hydrogens went, the electrons went too. All right? So we know that molecule B is the one that was oxidized. Okay? It was 2 electrons were pulled away from it. We know that because those 2 hydrogen ions were pulled away, and we see a reduction on the other side. Okay, so 2 things to keep in mind when you see these kinds of reactions. Always remember, in cells, redox reaction means that an oxidation and reduction are occurring essentially simultaneously. If you have an oxidation, you almost always have a reduction of some sort. Okay? So if that's the case, and you see the reduction on the right side of the equation, then all you have to do is look for the which molecule lost the hydrogen ions, and that's the one that lost the electrons as well. Okay. All right, this figure. Oh, any questions up to this point? You may be so confused by now that you don't know what to ask. Yes?
So is the oxidized, whatever's going to be oxidized, is it normally on the left side of the equation?
Yeah, just for our standard operating procedure, when I say which molecule was oxidized, I'm referring to the one that is on the initial reactant side of the equation. Okay? And then the products, it's already in the already oxidized form. So I'm asking which one was oxidized. It would be the one on the left. Yeah. Yeah. The question was asking, how do you decide which one it is, the one on the left or the one on the right? And it's the one on the left because the one on the right has already been oxidized. So the left one, the reactant, is the thing that gets reacted, okay, or broken down.
If you don't know what to ask, the study session is the place to be.
Study session is one good place to be, yeah. And the other thing is, if you haven't been using that CD-ROM that comes with the book, it's really helpful because there's a lot of practice exams, there's a lot of exercises in it to get you through these things step by step. Okay, so take advantage of that. What? If you use it, you can't return the book. Yeah, that's true. Okay, here's a summary. Well, yeah, exactly. You're only going to get $10 for the book anyway, so why sell it back? I used to sell back books, and I don't bother anymore because I never get anything for them. All right, so this summary shows you the three processes in our cellular respiration. Glycolysis, which is happening out in the cytoplasm. Glucose is being broken down by several steps into pyruvic acid. That generates a little bit of ATP, but that ATP is actually going to be used in the eukaryotic cell, so we can sort of discount it. We get high energy electrons carried by NADH. Okay, we get a couple of NADH molecules out of this. There is a step in between the glycolysis and KREBS cycle that we call grooming, which I'll describe, which pulls off a carbon dioxide and generates some more high energy electrons. The KREBS cycle generates some ATP. It generates a lot of CO2, and it generates high energy electrons. So all of these high energy electrons that are coming out of these prior processes then go into electron transport in the mitochondrion, and that process generates lots of ATP. Okay, note the Krebs cycle also occurs in the mitochondrion. It occurs in the matrix of the mitochondrion. The electron transport chain is happening in the inner mitochondrial membrane. Right? And then glycolysis in the cytoplasm. All right, as promised, I've got a flow chart I want to go through of this process. This is going to be really your starting point for studying. Most of what you need to know is right here on this flow chart. Right? So you want to really pay a lot of attention to this when you're studying this for the upcoming exams. We start with glycolysis. Right? It is the process of taking a glucose and breaking it down into 2 pyruvates. You're taking a 6-carbon compound, you break it into 2 3-carbon compounds. You haven't lost any carbons, but you've done a lot of bond rearrangement, as we'll see when we go through the process in detail. All right, so glucose to 2 pyruvates. That generates 2 NADHs and 2 ATPs net per glucose. Okay, we actually generate 4 ATPs, but 2 of them are used. Right, so we generate 2 NADH, 2 ATPs. All right, we're going to sort of forget about those NADHs and ATPs for right now because we're sort of done thinking about glycolysis. If oxygen is present, right, then the ATP and the pyruvate are brought into what we call a grooming step, which I haven't talked about up to now. This grooming step does several things. It pops off 1 carbon from each pyruvate as carbon dioxide, right? We exhale that. It generates a molecule called acetyl-CoA. These acetyls just have 2 carbons each. Okay, so from the 3-carbon molecule up here, we generate 2 carbon molecules, 2 CO2s, so we still have all of our carbons accounted for. And again, high energy electrons because we've pulled off those carbons and captured the electrons. Right, so 2 reasons cells do this. One of the reasons that they do grooming is because it generates NADH. It allows them to capture high energy electrons. The other reason is because it's required for entry into mitochondrion. You can't get this stuff in the mitochondrion unless it's bound to this cofactor called coenzyme A. It just won't go in. Okay? So you have to get it into the mitochondrion, and so you do this reaction to do that process. Once in the mitochondrion, the acetyl-CoAs enter the Krebs cycle. All right? So we're going into the Krebs cycle with these acetyl-CoAs. These CO2s have been exhaled. The NADHs, we're going to forget about them for the moment. We'll come back to those. Okay, the Krebs cycle goes through a number of steps, but basically what it does, it takes those acetyl-CoAs and produces 4 CO2s. All the carbons have been broken down to CO2 at this point. 6 NADH, 2 FADH2, and a little bit of ATP. Right? 2 ATPs per glucose that started out in glycolysis. The big payoff, though, is these two molecules, the NADH and the FADH2, because those are high energy electron carriers, and you get much more ATP by using those in electron transport than you do by just generating ATP. All right, so we have all of these carriers of high energy electrons. NADH from glycolysis, NADH from the grooming step, NADH and FADH2 from Krebs cycle. All of those go into electron transport. This step, however, and I should have indicated this in this flow chart, but I didn't. This step, bringing the NADH into the mitochondrion, takes 2 ATPs. It requires 2 ATPs. So you might want to add that in. Okay? This 2 NADH requires the use of 2 ATPs to get it into electron transport. All right? So that's why I say that we can pretty much discount the ATP that's generated in glycolysis. We make 2 ATPs, but then we use 2 ATPs to get the NADHs in. All right? So there's really no net gain there. All right, in electron transport, of course, you're taking those high energy electrons, passing them off to electron carriers, passing them down an energy gradient, pumping hydrogen ions, letting them flow back through, and you generate ATP. A maximum of 34 per glucose. The number of ATPs changes according to physiological conditions. Right? If you need to get warm, then you generate less ATP, because what happens is your body starts allowing those hydrogen ions to flow back without making ATP, and it releases heat. Okay, so you warm up. So the maximum of 34 ATP per glucose. So what do we have here? We made 2 ATPs in glycolysis up there, but we lost those when we transported NADH into the mitochondrion for electron transport. We've got 2 ATPs here, 34 ATPs in electron transport, so a maximum of 36 ATP per glucose if oxygen is present. Where did you get the other 2? Where did I get the other 2? Right out of Krebs cycle. Right there. Yes. Okay, so obviously it's a fairly complicated process if oxygen is present, but boy, the payout's big compared to when oxygen is absent. Now, not all cells can do this. Not all cells can operate in the absence of oxygen. Some actually are poisoned by oxygen and cannot have it around. But most cells use oxygen. There are some cells that will operate, however, in the absence of oxygen. If they do so, then they go through a process called fermentation. They don't go through any of this other stuff, Krebs cycle or electron transport. Instead, they take these pyruvate molecules and they put them into one of 2 different pathways depending on the cell type. Okay, one of the types of fermentation that's done is called ethanol fermentation. Okay, you take the pyruvates, you break them down into CO2 and alcohol. Okay? Without this, we couldn't have beer and wine and other fermented drinks. Okay, yeast cells will do that pretty efficiently. Another process is lactic acid fermentation or lactate fermentation, where you take the two pyruvates and you just change the bonds around a little bit to produce a molecule called lactate. Our muscle cells will do that anaerobically. Right? So if you're sprinting full out from a dead stop, your muscles haven't had a chance to get enough oxygen, and so they're doing this anaerobic process to try and generate enough ATP to keep the muscle contractions going. After a while, they catch up and they're able to do the aerobic process. Okay. Why do cells do fermentation? The reason is because in glycolysis, you generate NADH, you generate ATP. Okay, that's well and good. The problem is you can't pass these electrons off anywhere. Okay? There's nobody to pass them off to because they're not going into electron transport. There's no oxygen. So what happens instead is you have to take these electrons and pass them off to the pyruvates again. Okay, that's what's happening here. You take these NADHs, you pass the electrons off to pyruvate, and you generate either ethanol and CO2 or lactate, depending on the cell type. That regenerates NAD, so you can go through glycolysis again. And so that's why cells do it. If they didn't, they'd run out of NAD and FAD. Okay, that's a good place to stop. So let's stop there before we start getting into the details of the process. And I'll review this a little bit on Friday as well, just so we're straight on it.